It's a great pleasure to give today this talk. This is a joint work with Timo Close, who is in Botany and Plant Sciences at UCR. And this is work done mostly by my student, Hamid, that could not be here and present his talk. Too bad. So the context of this work is in the domain of ultra-deep sequencing. So with ultra-deep sequencing, we mean sequencing which has a coverage of at least 1,000x. And you might think, well, this is probably very, very rare. It turns out that we found you can find at least a half a dozen papers already, this one down here, that use this type of coverage for real experiments. And I think that this will happen more and more often because sequencing costs keep decreasing. And so this work is sort of the core question is, what if you could sequence everything at no cost? Would you sequence as much as you could? And what is the consequence of doing this? So we tried the following experiment about a year ago. We sequenced several bags with a very, very high coverage, 8,000 to 15,000x. And we tried to run assembly algorithm, de novo assembly algorithm on these data sets. And the results were very poor on the whole data set. And so we said, well, let's try to see what happens if we cut some of the data out. So we plotted these statistics, assembly statistics, common assembly statistics like M50, the longest counting, and the errors, and so on and so forth, for increasing coverages, right? So you have 2,000x, 4,000x, 6,000x, and so on and so forth. And what you would think, the expectation would be the more data, better assembly, and this is true initially. So as the coverage increases, so this is a full coverage, all the statistics gets better and better, but something interesting happens after you go above 2,000x. The actual quality decreases. And all these data, by the way, is reported in this work that appeared in Bioinformatics about a month ago. So this is real data, this is Velvet, and we observed essentially that there is some sort of a sweet spot around 2,000x that gives you the best sort of trade-off in terms of amount of data versus quality of the assembly. So this is, again, de novo genome assembly. These are short bags, 150,000 base pairs long. So that was interesting, and we were still puzzled by these experiments. We tried this on Velvet, we tried DVA, we tried Spades, and they all show similar behaviors. More data does not mean necessarily better assembly. It is true at the beginning, but at some point it's not true anymore. So why? Well, we still don't know. But through simulations, we listed a list of possible suspects here, like sequencing errors, amplification biases, PCR amplification biases, chimeric reads, and perhaps even the fact that these assembly algorithms were not designed with this very, very high coverage in mind. And maybe some of the internal parameters need to be tweaked. Nonetheless, these happens. And we eventually were able to identify that sequencing error may be one of the main causes of this by doing simulations. So we showed that if the reads are error-free, you don't observe these. As soon as you inject sequencing error, you start observing this degradation of the sequence of the assembly quality. And again, you can find this study in this paper here that is recently published. So how do we deal with this problem? So if sequencing errors are the cause, then perhaps we could correct the reads. And typically, error correcting tools are based on rare k-mers. And it turns out, and I will show you some results later, that they're actually ineffective for this kind of coverage, so very high coverage. The other possibility would be to simply take a piece of the input and just run the assembly on that fraction of the input. And this is, of course, not easy to do. There are some techniques, but it's actually not a very satisfactory or intellectually interesting idea in the sense that we don't know what we're going to throw away. Maybe some of these reads are actually might be useful for the assembly. So what I'm going to present next is a different approach. It's actually a relatively simple idea that we call slice assembler. And you will see in a moment why it's called that way. So the idea behind slice assembler is divide and conquer. So since we realized that you could get better assembly by by cutting them, like cutting the data into pieces, that's what we do. So instead of running the assembler on the whole data set, we cut the input into n slices so that the size of each slice is optimal in the sense that maybe it's 1000x, 2000x, 800x, whatever we think is actually the appropriate coverage that will maximize the quality of the assembly. So once we have cut the input into slices, we run the assembler, the base assembler. It could be Velvet, IDBA, Spades, your favorite assembler on each of the slices, okay, where each slice is a piece of the input. And then we end up with, obviously, n assemblies that needs to emerge. So this is step 2, the assembly. In step 3, we look at these assemblies and we try to find the good part, right? So these assemblies are expected to contain good stuff and bad stuff. And so in this step, in step 3, we try to find what's the good stuff. And so we have n assemblies, and in order to find what's good, we take a majority vote. We look at what is common to the majority of this assembly. Well, it's a very simple idea. So we look at for these frequently occurring substrings, or FOS. So these are substrings. It could be the whole contig or a fraction of a contig that appears in at least, say, half of the assemblies. Okay. And in order to do this efficiently, we use the generalized suffix tree, as I'm going to explain next. So an FOS for us is a substring that is maximal, that is at least L nucleotides long, and it appears in at least K assemblies. Okay, so initially you can say K to be N, but then you might want to relax K as you go along if this is too stringent. So in order to do this, this problem is a generalization of the longest common substring problem to n strings, and it can be solved, for example, using a suffix tree. You can build a generalized suffix tree and then color it, so where each string has a different color. And the question is to find, so here's a picture of a suffix tree. In this case, you have 3 strings. And the objective is to find these maximal substrings by annotating the suffix tree with a number of colors in the internal nodes, and then looking at strings which satisfy the length requirement. So, again, we want to find the maximal ones to reach a number of candidates, and in order to do that, we use the suffix links, but this is technicality that can be found in the paper. Everything can be done in linear time, which is nice. And once we have these FOSs, remember these FOSs are the common substrings to the majority of the assemblies, we merge them. We merge them not only between themselves, but also with the FOSs that are found in previous iterations. And this is an iterative process, and I'll explain in a minute how this is iterated. But in this step, we merge FOSs, these long common substrings, together by doing prefix-suffix overlap. So in order to be very conservative, we expect the prefix-suffix overlap to be exact. And we also expect the prefix-suffix overlap to be of a minimum length. And if the length is not sufficient enough, we look at parent reads that can confirm that overlap. So we try to be as stringent as possible and not to make any mistake in assembly. Once we have, as we're building the consensus assembly that eventually is a set of all the FOSs, a consensus assembly, we are going to do another round. In order to reiterate process, we remove the reads from the input that are present in the current consensus assembly. Right? So the idea is that we don't want to reassemble the part of the genome which has already been assembled, which is already in the consensus assembly that we're building. So we get rid of them. And this actually makes the problem of assembling easier for the assembler, so they can concentrate only on the regions of the genome which has not been assembled yet. So in this step, we use BWA to filter out all the reads that are mapped precisely into the FOSs, the current consensus, remove them, and then we iterate. Right? So we go back to this step, and then we assemble again, find the FOSs, merge them, and so on and so forth, and this process continues. So it eventually stops, and it stops when we cannot find FOSs anymore. So there is nothing in common anymore between these assemblies. So how about the results? So we had 2 sets of results on real data and on synthetic reads. On real data, we have sequenced on the HiSeq 2000 16 Barley bags. Barley is a genome which is 5.3 billion base pairs, very repetitive, very hard to assemble. And we've been working on Barley for a long time. But we selected 16 bags to be sequenced at super high cover, 8,000 to 15,000 coverage, using parent reads, which were at 88 base pairs after trimming. For consistency, we use 8,000x coverage for the bags. And out of 16 bags, 5 of them we had the Sanger-based high quality reference assembly. So we could use that as a ground truth. For synthetic reads, we used WGSIM, and we generated data at different level of coverages and rates to experiment and fine-tune the assembler. So here are some results on the 5 bags for which we have a ground truth. The 5 bags are here at the bottom, 1, 2, 3, 4, 5. And we have 4 experiments. We're running Velvet at 8,000x. We are running Velvet after Racer. Racer is an error correcting tool on the whole data set, again, 1,000x. We are trying also to run Velvet on a tenth of the data, which is the size of the slice, 800x. And in blue, we have Slice Assembler, our tool, using Velvet as the base assembler. Slice Assembler is not an assembler, it's a meta assembler. It uses an assembler inside and iteratively processes the output. So Slice Assembler uses the whole data set. And as you can see, if you look at number of contigs, longest contigs, and 50, and so on and so forth, Slice Assembler actually does a very good job. In fact, with the exception of back4 in this panel, Slice Assembler is actually the best tool among all these 4. And I want also to point out that the number of errors is extremely low. So Slice Assembler is extremely conservative and accurate in producing the assemblies. You might wonder why Velvet and nothing else and something else. Velvet is not the most recent assembler. It works very well on bags, but for your curiosity, we also tried IDBA and Space and Ray as the base assembler. And in all these cases, Slice Assembler in conjunction with that base assembler has actually always improved the quality of the assembly. So again, in this case, the base assembly is run on the whole data set, but Slice Assembler is run with that same assembler, but on 10 slices of 1/10 of the data. And as you can see, most of these statistics, if not all, like in 50, the longest contig, and so forth, they're all improving. On simulated data, we play with the sequencing error, since we thought that sequencing error was the culprit in the degradation of assembly quality. So in this case, we generated reads with increasing sequencing error rate. So at 0%, you have perfect reads, and at 2%, you have super error plagued read. And here we are comparing Velvet, naked Velvet, to Slice Assembler plus Velvet. And again, Slice Assembler plus Velvet actually does a better job in almost all these statistics. And again, very notably, produces no misassembly of any kind on these assembler, even if the input reads are 2% of sequencing error, which is quite remarkable. Okay, so I'm to the conclusions. I have presented a meta-assembler that is in response to the observation that modern de novo genome assembler seems unable to take advantage of ultra-deep coverage. So Slice Assembler is take advantage of the whole data set, and because it's using this idea of majority voting, is more resilient to sequencing error than its base assembler, and very rarely introduces misassembly in the assembler, in the final assembly, which is very good. Slice Assembler is available as a software tool right now. It's actually is a collection of scripts, and we are implementing this in C++ and eventually release it in a few months. Slice Assembler is low because it's an iterative process, and it calls Velvet on all the slices, or whatever is the base assembler, can be 40, 50 times lower than the base assembler. So we are working on ways of making it faster and being able to scale to larger genomes. With this, I want to thank the funding agency that supported this work, and thank you very much for your attention.
Thank you very much. Any questions from the audience?
Good afternoon. It was a brilliant work, I would say.
Thank you.
My question is, I have a couple of questions actually. Towards the end of the slides, you said your misassemblies is very low or kind of negligible. So how are you defining misassemblies?
So in the first part of the work, we were using the gauge script to compute misassemblies. So it was, I think, indels of at least 5 base pairs and miss points. In the second part, we're using Quast. So we have, because we have the reference, right? We have these 5 high-quality Sanger-based assemblies. We can just use Quast and compute. It's the number of misassembly reported by Quast, essentially. So that's the answer.
My second question is regarding the sequencing errors. Now you have used Velvet as your base assembler. But, as far as I know, Velvet also has its own error correction, though very not
primitive. Right.
So did you see the effect? Like, is it is your error correction thing due to your assembler, or is it due to Velvet's error correction algorithm?
No, but yeah. So most of the assembly algorithms have a pre-processing phase in which they try to correct the errors. No, I think the reason we don't produce any misassembly, very very rarely in the final consensus assembly, is because we are very very careful, very conservative on what pieces of those assemblies we're picking. Right, and so it turns out that our choice, we're by being very conservative, we don't make any mistake. And so we hope that this can extend to larger genomes. These are bags, 150,000 base pairs long pieces of DNA. So but they come from a very repetitive genome. So yeah.
Thanks a lot. Brilliant.
Sure.
Do you think any of your approach is applicable to metagenomics?
Yeah, metagenomics is a domain in which you do have a very, very, very high coverage sometimes. Right? These are relatively short jumps. We haven't tried it, and I'm not sure. So you're thinking about metagenomic assembly? Yeah, I don't know. Yeah, that's a good question. Another possible area of applications would be co-assembly, where you have single-cell sequencing of multiple cells, and then you get multiple assemblies, and you want to merge them together. Right? But we haven't tried that yet.
So I was wondering whether you as you say, this is likely about the assembly algorithms. In fact, I know the assembly algorithms make assumption of the coverage range, in particular about the error profile. Did you consider going and changing the error correction routine inside of Velvet? I mean, it's right there to be changed, and that might actually be a simpler thing, is just to change the way the error model. The assumptions of Velvet about how sequencing errors distribute. In particular, Velvet makes the assumption, I'm pretty sure, Daniel's not here, I'm pretty sure Velvet makes the assumption that errors are unique in the thing, and obviously at 8,000x, that's not going to be the case.
That's right. And that's why Eraser also doesn't help. So I agree. So we could mess with the internals of the assembly, the assembler, but and the same is true for Spades or IDVA, or they always have a pre-processing which try to correct. There is. We decided not to touch them and just you have a wrapper around it and see if you can get the best out of these. Yeah. Okay. Good point. Yeah.
Okay.
Thank you.
One more question over there.
Hi.
Hi.
I found that digital normalization is extraordinarily effective at improving assembly quality in ultra-deep viral data. Have you explored that at all? Have you paired your tool with it?
We did look into it a little bit, but then we decided to try to see if we can advantage of the whole data set without trying to remove anything. I mentioned this in the sampling approach. It's a possibility. It's an alternative. But we found that would be more. It would feel safer to try to use the whole data set and not take the risk of removing reads that might actually be useful. So I'm sure that Diginome is a great tool, but I don't know what it does internally, and maybe sometimes it makes a mistake and remove a read that is actually useful, and maybe it will bridge two contexts, and I don't want to get rid of it. So that was the idea. Yeah. But thanks. That's a very important point.